Awesome. Hi, everybody. Really great to be here. Really enjoyed the talk so far. To just set the stage and get some information from you guys, will you raise your hand if you would consider yourself very familiar with cultivated meat? Okay, great. A lot of people. Awesome. Okay, I'll try to dive a little bit more into the details then, so it's not just an overview. First, just a shout out to Tufts University, my home institution that I'm very, very proud of and just so excited about what Tufts is doing in cellular agriculture. So the Tufts University Center for Cellular Agriculture, which we call TUCA for short, is basically the umbrella of just all the cell ag activities that happen at Tufts. It was officially established in 2021 when the USDA granted $10 million to the university to establish an interdisciplinary research program. And it's really just exploded ever since, really driven by enthusiasm from students. We see just this huge driving force from how many students are just attracted to this technology and really want to build a workforce. And so Tufts has really tried to respond to this by building educational resources to help the students, go down the career paths that they're really excited about. So specifically, some initiatives that we were able to develop. We have a certificate program, so you can go to Tufts and get a certificate. This is at the graduate school level to just add that accreditation in this new field. And we also recently have a Cell Ag minor program where undergrads can get a minor in cellular agriculture. We've developed 4 new courses in Cell Ag so far, and all of the course materials are online for other universities to build off of or access if they're interested. We obviously have a lot of research going on at the university as well. Today there's over 50 active researchers. Most we're really heavy on the cultivated meat side of things across 5 research groups and a number of publications. And then the pillar that I work really heavily on is innovation. So I founded and I run the Cellular Agriculture Commercialization Laboratory, which is a new group within our innovation office that works on translating these technologies from the lab to market. As Dr. Messer mentioned, you can do it, but is it viable? And that's a really good question and one I'm becoming intimately familiar with. I did my PhD at Tufts and was very heavy on the academic side, and for a while was a very build it and they will come kind of mindset, and I've really adapted to the other side of the spectrum, so that resonated with me a lot. And then Tufts also has an industry consortium, so we work with industry partners to inform priorities for pre-competitive research. We were talking a little bit about definitions, and I guess I like laying out this framework for how I think about things and talk about things, since we use a lot of different words for sometimes the same things. So obviously the overarching goal is to replace conventional products that aren't produced very sustainably, usually because they're from animals, with alternatives. And we have both foods and materials that fall under this goal, and there's similar approaches to products in both of these categories. So obviously plant-based and fungi-based and algae-based. I didn't even cover all my bases because we have insect-based and bacteria-based approaches as well. Are kind of one half of the equation where the goal is to produce analogs ingredients. And then we have this cell-based category, the cell-based approach. And the 2 subcategories I have in there are byproducts from cells or the whole cell itself. And how I think about this being different is rather than creating analog products, we really are trying to create the exact same product from the same essential ingredients, but doing it under more sustainable production methods. And so these red boxes are what we call cellular agriculture in juxtaposition to animal agriculture. And of course, the byproducts are the precision fermentation approach, and the whole cell category is the cell cultivation and tissue engineering approach. And we most commonly think about cultivated meat and poultry and seafood falling under this category, but there are also interesting materials applications that use a similar approach. Leather is kind of an obvious one, but I think even like non-animal cells fall in this category, at least in my head. And there's a company in Cambridge that's creating lab-grown wood, which is whole wood cells to create alternatives to conventional wood products. So there's kind of, can think outside the box with new categories as well. And Taka and me specifically, I just really focus on cultivated meat. I'm absolutely obsessed with it. I found out about the concept in college, and just in a second, I was like, that is what I'm doing for the rest of my life. That's the coolest thing I've ever heard of. And 10 years later, I've never been bored a single day. It's the most interesting, exciting field, and I love talking about it. So a little bit of historical context, because I think we're at a really interesting place in the evolution of the industry right now. This is a chart that's probably familiar to a lot of people, how technologies evolve over time with regards to the public's expectations of the impact they'll have. And I think we can kind of all realize that cultured meat originated in a concept of science fiction. So it was first, described in science fiction books like The Space Merchant from the 50s, and it was depicted as replicator meat in Star Trek. And everyone kind of quotes this Winston Churchill quote that he wrote in an essay in 1931 where he was predicting what the future would look like in 50 years, and he had a quote about basically depicting cultured meat. I like throwing up this lesser-known quote by, I think it was one of Winston Churchill's friends. I think he said it like a year earlier, and he also had a more accurate timeline. He wrote this quote in an essay called The World of 2030, so we could still make that timeline. We could still potentially commercialize these products in a meaningful way by 2030, so I like using this quote. And I'd say this, period of time roughly lasted through the 1990s. Because in the early 1990s, people actually, were inspired by this concept and started taking meaningful steps to actually make it a reality. So the first patents were filed in the 1990s. NASA actually funded what I think is the first public research on cultivated meat back in 2004. They were looking at cultivated meat for space food as an application. They actually grew goldfish meat. The photo of cells is the goldfish cells they were able to propagate. And then, of course, one of the first studies was from Hannah on the environmental impact in 2011. And there's actually an old economics study, which is interesting to look at now that we have more updated TEAs that was published back in 2008. So just interesting to kind of look back. It's not that long ago, but our thinking has really evolved. So that's what I'd call the early research state of the industry. And then between 2011 and about 2023, at least in my today's perspective, I'd say is when the industry really boomed. So over those approximate 10 years, there was over 170 cultivated meat companies that popped up around the world. I was in graduate school, and I remember we used to be able to really just count on one hand the number of companies that existed, and we saw a new company existed or popped up in the news. It was a huge deal. And after a couple years, we couldn't keep track. There's just so many efforts really proliferating around different products. And then I think, a few of the just big wins that really mark, this industry boom period was Good Meat getting regulatory approval in Singapore to actually sell cultivated meat. That was in 2020. And then in 2023, two companies received approval both to sell cultivated chicken in the United States. So at least with today's current perspective, I'd say that was kind of the height of the industry, or this one peak. And pretty shortly after, I think industry collapse is a little dramatic. I don't know if we're quite there, but pretty soon after these approvals and these really, really huge milestones, we saw a huge pushback, and there was suddenly a lot of negative articles about cultivated meat, that it was overhyped. There's been bans, obviously, in the United States, and also, I believe, Italy, passing bans on cultivated meat. And then in 2023, the same year that we had these regulatory approvals, new company formation investments declined by 75%, which was really just notable in the industry. And then this year, multiple companies have shut down or announced major layoffs. So we're perhaps at this trough, but rapidly coming out of it. I'd say, like, moving forward, we're in a period of ecosystem building. I think the first, era of this industry, like Joshua was talking about, was a little bit competitive, a little bit siloed, and now we're kind of learning from those experiences and realizing we have to be more collaborative and not just build individual companies or individual efforts, but really specialize along the value chain and more ecosystem building. And some really positive signals, among many others, that I'm excited about are, $100 million in funding from the Bezos Earth Fund to fund these sustainable protein centers at Imperial in Singapore and North Carolina. I think those will just be really instrumental, obviously, for cultivated meat and the other alternative protein categories for pushing things forward. I saw that recently there was regulatory guidelines published by South Korea, so that will be a great resource. And I saw Nutreco is opened like a cell feed production facility, something that didn't exist before to specifically generate cell culture media for cultivated meat. And then a Tufts-specific positive signal, there's also been a lot of talk in the industry about the need for shared infrastructure and how it's so expensive for each company to invest in its own CapEx. And so one of the ways we responded to that, we got $2 million in funding from the state of Massachusetts to develop a shared scale-up pilot facility that academics and industry can access bioreactors up to 50 liters to start on that pilot scale production journey. Okay, so now going back to the technology and diving into some of the science. So I think everyone's very familiar. This is the general production process for cultivated meat. So I'll just talk about each of these in a little bit of detail. So the key ingredient for cultivated meat is obviously the cell lines. That's what gives the meat its species specificness and what really dictates how your bioprocess is going to look. And we do get these cells from animals, obviously. There's a lot of different types of cells that you can build your process off of, and there's different reasons for, pros and cons for each approach. You could use embryonic stem cells, which are really great at growing and getting a lot of biomass. It's a little harder to tell them specifically what cell to become. So if you're trying to create muscle, you might not get a super high conversion rate to muscle. There's also induced pluripotent cells, where you can get a cell from an animal very non-invasively and reverse engineer it to become a stem cell, and then set it down a maturation path towards muscle or fat or whatever you're trying to create. And then there's also primary cells, where you get cells from the animal that already want to become muscle cells or fat cells. So they have some predisposition to become what you're looking for. They don't grow quite as well as some of the other cell types. So there's just trade-offs depending on your process and also just what you have access to. At Tufts, we primarily, I think just because of the expertise we've had on hand, work with primary cells. So Tufts has a veterinary school associated with our university, so they raise a bunch of livestock. So our students will go to the farm, work with a vet, get a health certification for the animal. We have, obviously, the IACUC approval, so there's a lot of documentation going into that already as we think about food safety and just getting these processes in place for later stages of the process. And they anesthetize the animal, and then we get either a little piece of muscle tissue or a little piece of fat tissue. It's about the size of a jelly bean, so it's really small. And we'll put that in growth media and drive it back to the lab and isolate the cells within that piece of tissue that can proliferate and become the type of meat that we're trying to grow. So this is what our cow muscle cells look like under the microscope and stained. I think obviously the coolest image is the lower right-hand image, which is muscle tissue grown in vitro that has been differentiated. So you can see all the cells have fused together. The blue nuclei, or the blue staining, is cell nuclei, and you can see that's all in one muscle fiber. And then the red staining is those muscle-specific proteins. So we can treat the cells in different ways and measure how well they're actually turning into muscle fibers. And it's really cool to see under the microscope, just like your actual muscle contracts, these cells will contract in cell culture, which is pretty crazy to see. A few other cell types that other people are generating that we're excited about. The Roslin Institute, Roslin Technologies, has a pig fat cell line that was spontaneously immortalized, so no genetic engineering to get it to be a really good growing cell line. And I'm pretty impressed by how well the cells can accumulate lipids. So over 40 days of differentiation, you see like that big green balls. Those are lipid droplets within cells. And that's a really impressive amount of lipid accumulation that could, start to emulate what we see in vivo pig fat. And I believe this is a cell line that people can access for R&D applications. And then just another example of cells that people are growing. We tend to do a lot of muscle and fat, but fibroblast is also another good option. They grow really well, and then you can differentiate them into muscle or fat tissue. And that's the cell type that Believer Meats is growing. And they have a couple of publications that describe their process in good detail. Okay, so the culture media, another very important part of the process. So this is the nutrient broth that is the feedstock that feeds the cell lines in the bioreactor, and is a huge, field-wide area of priority for research because it's at least has been the main cost contributor driving the high cost of cultivated meat. And this is largely because, we got these formulations from the pharmaceutical industry and we've slowly been adapting them, but we were just using inputs that were designed for a very low volume, high margin industry, and now we're trying to flip that on its head and produce a lot of volume for low margin in food. So it will just take a while to adapt these inputs towards something that will make cultivated meat something that we can achieve price parity for. So this is a example formulation of a culture media recipe. A lot of people know that fetal bovine serum is something that's used in cell culture. I do think the industry is at a place where serum-free is very feasible. Usually if you're starting to get a new cell line, maybe a species you haven't isolated before, you will isolate it initially in serum because it just works so reliably. But now there is a lot of resources and literature out there that kind of make it easier to move to a serum-free system. And there's like the cow, pig, chicken cells have been definitely validated in serum-free media pretty extensively. So this is what a serum-free media recipe looks like. You have amino acids, sugars, recombinant proteins, and vitamins, growth factors. And by looking at the cost, we can see very clearly within the growth media, recombinant proteins and growth factors are driving the vast majority of this cost, and most of it really being these recombinant proteins. And these are proteins like albumin and insulin and transferrin that are very prevalent in the media. So one of the research projects that my colleague Andrew Stout performed at Tufts was to both develop a serum-free media recipe for the cells we were using, and then also work on decreasing the cost through tuning the ingredients. So this is just a graph of how we've been able to decrease the cost of culture media from the initial baseline, which is growth media that contains fetal bovine serum, which can be upwards of $250 per liter, and you need many liters just to create 1 kilogram of meat. So obviously you can see where the high cost comes from. He was able to develop a completely serum-free version of this growth media, and then also drive down the cost by just sourcing larger scale components, and then also reducing the amount of growth factors that are needed. And so when he kind of arrived at his final recipe that works for cow muscle cells, he saw that the biggest cost contributor of this media was recombinant albumin. And that's because it's so prevalent in the media. I think about like 90% of the bulk of the recombinant proteins in the media is this albumin protein. It's also the most prevalent protein in blood, so it makes sense that we would need it in culture media, but it was really driving the cost and making it very difficult to reach price parity with conventional meat. And so his next project was to find an alternative for this really expensive ingredient. And I think this is just a great signal that it is possible to reduce the cost if we're just thinking about media optimization in ways that pharma hasn't thought of it before. So he developed a new media called Beefy R, where he was able to replace recombinant albumin with a byproduct from canola oil production. So this ties a lot into the Protein Innovation Center work. We did a lot of extraction, optimization, and solubility to get this to be something that would work well in cell culture. But basically, the byproduct of canola oil production is the rapeseed meal, and we're able to isolate an albumin-like protein from that and replace it 1-for-1 for recombinant albumin. And already at lab scale, it's like 40 times cheaper, and we should be able to get to $10 a kilogram of this protein, whereas recombinant albumin is on the shelf for about $40,000 a kilogram. So this is one meaningful step we can take to reducing the cost. And then I'll just quickly go through the other things. Scaffolding is another component of the process. I'd say it's more optional. You can create cultivated meat without scaffolding materials. You could just grow a lot of cells in a bioreactor, collect those cells, and then process them into a meat product. Scaffolding comes into play when you want to create something that's structured and actually maybe resembles a whole cut, or maybe has both muscle and fat, and you see patterning and organization, and that's when you'd want to use a scaffold. And there's a lot of different materials, plant-based materials that people can use for scaffolding. I'd say one of the big challenge areas is getting cells to adhere to non-animal based materials. So cellulose would be, really great if it works because it's abundant and cheap and edible, and people, recognize that as something that's in food, but cells don't love to stick to it. So there's still a lot of innovation in scaffold development. Quickly going through bioprocessing, so some just random figures here. I think one really important part to think towards bioprocessing is getting cells that grow in suspension culture. A lot of the cells that we do research on are adherent, and that's a very hard process to scale because people have not designed equipment that can propagate adherent cells at the volumes we would need to make cultured meat industrial scale production. It really is suspension systems that already exist where we can reach scale. So getting cells to grow in suspension culture, I think, is really important, and something that Believer Meats, again, has done, and how they adapted their cells to suspension culture in these aggregates, and we're able to get something that can grow in equipment that already exists today. And then just this, I like kind of seeing where we are today in terms of production capacity. So I liked this survey output from the Good Food Institute that surveyed a number of companies in the industry and asked how much they would be able to produce by the end of 2026. And the aggregation of the production estimates from 19 companies estimates that the industry will be able to produce the amount of meat that currently comes from one US slaughterhouse by the end of 2026. So not a whole lot, but a lot more than we've been able to produce in the last few years. So definitely a mark of progress. And I think I might, I have a couple more minutes. I guess I'll talk about some of the sensory studies. So I'll skip to aroma. This again was a study that came out of Tufts University, one of the first sensory studies. For a while it was just hard enough to produce enough sample to do sensory analysis on, and also to get the approvals that you need from an academic institution to actually have human sensory panelists. But we recently published a sensory aroma study on cultivated pork fat. So people smelled cultivated pork fat and conventional pork fat and listed the flavor notes. And then we also did GCMS to measure the volatile compounds. And we saw similar, but not 100% the same, of the types and amounts of volatile compounds. But I think a really good sign is that when we did the sensory panel, there was a pretty good alignment between the flavor notes that people were able to smell, and people couldn't really tell the difference between, or they couldn't tell which one was cultivated and which one was conventional, which again is just a good sign. And I think I'll skip to the end for, I don't know why it's jumping around. Yeah, so just to go over some of the challenges, I think one of the biggest challenges that's really worth collaboration to focus on is just cell line development, because it's such a key research input. Right now, a lot of companies will develop their own cell lines in-house, and that's an effort that costs probably between $2 and $20 million, and each company is developing somewhat similar cell lines and then using those just as a proprietary piece of IP that's hard to let go of. So one of the initiatives that we're really trying to establish at Tufts is the idea of an open access cell bank, where we can bank cells that are available for R&D use and commercial use that don't have any inhibitory economic constraints. So these would be free from royalties or inhibitory fees, but able to be bought as a research input by companies and academics to help their research, and also make sure that people are studying the same cell lines that people are actually producing food from. And yeah, for sake of time, I'll stop there.
Thank you very much.